I guess we can move to the next session. Good morning. My name is Domenico Praticò. I'm a professor of neuroscience at the Temple University in Philadelphia. It's such a pleasure to be here with you today. I'd like to, first of all, also recognize the organizing committee, the chair, for putting together such an exciting and very stimulating program, and particularly for inviting me to chair a session which is a little bit different. We are going to switch gear. So far we heard about all the benefit the Mediterranean diet, olive oil has on cardiovascular, metabolic, cancer. Now we switch. We move a little higher, north. We move to the brain, which is wonderful. As you know, the old Romans said, mens sana in corpore sano, which means you have a good brain if you have a good body. So the two things are very well connected. So the benefit on the cardiovascular have to reflect the benefit in brain. But it's really exciting and a great pleasure, honor to introduce our two speakers today. I will start with Professor Mary Yannakoulia. She's a professor on nutrition and heated behavior in the Department of Nutrition and Dietetics in Harokopio University in Athens. She has been very productive and in an area that's very, very important, which is the influence that eating behavior, lifestyle, diet, nutrition has on cognitive function, cognitive decline. Dr. Yannakoulia has been involved in several international and national studies during her career. She got many awards, the Fulbright Research Award, and more recently also the European Dietetica Fellowship Award. She's going to talk today about the influence that nutrition, in particular focus on the Mediterranean diet, has on cognitive decline.
Okay, good morning to everyone. Thank you, Domenico, for the kind introduction. I would like to thank organizers of this meeting, Stefania and Helena, for inviting me to be here in this wonderful city, wonderful country, to meet all these people in this meeting. I really enjoyed these 2 days. So my presentation today is about nutrition and cognitive decline. I would like to give you the evidence in the next 20-25 minutes. Okay, I have no conflict of interest to declare. However, I must admit that I come from Athens, Mediterranean country. I practice Mediterranean diet, and this might be a conflict of interest according to John Ioannidis. Okay. So before starting, I would like to clarify 2 issues. First of all, I will focus on cognitive decline, but this is one of the physiological changes that occur during aging. There are also other physiological changes, like, let's say, physical weakness, that interact with psychological changes like depression and also the accumulation of disease. And in real life and also in research, you cannot separate all these changes that occur simultaneously. And these things need to be taken into account when we study human beings. And on the other hand, I will discuss how diet affects cognitive decline, this side of the direction. But again, in real life and also in research, there is also the opposite side of direction. We do not know that people, as they get older and they have more cognitive deficit, they also change their life because of their cognitive deficits. But I'll try to focus on this side of the direction, but this also has to be taken into account when we do research on human beings. So the evidence is huge. So I decided to use as a starting point the review we had 6 years ago, along with Nikos Karmeas. I know that many people in this Congress know Nikos from his work in neurology and dementia. So for this review, we had very strict selection criteria. We included only longitudinal studies and clinical trials, and longitudinal studies with a significant amount of participants, more than 1,000 amount of participants for longitudinal studies and 100 participants for clinical trials, and a minimum duration of half a year. We included studies where participants were healthy, cognitively healthy at baseline, and we focused only on clinical outcomes, not on biomarkers. These were our selection criteria. So I will update this review by including more recent clinical trials or longitudinal studies, some important ones, and also meta-analysis, and I will focus on food groups and Mediterranean diet, not on nutrients. So this is the kind of tables I want to present you. So you see here, oh, they changed the pointer, okay. Thank you, Ma. Oh, where is that? But I lost it. Okay, it's okay, I lost it. Okay, so here you see the food groups, and then the observational studies, the prospective studies, and then the clinical trial. Every dot represents the net effect found in the study. If the dot is green, the effect of this particular food is protective. If it's yellow, there is no effect, and if it's red, it's detrimental. So I will start with alcohol, okay, and there are no clinical trials, okay. But I think that the observational evidence indicates that moderate alcohol consumption up to 1 to 2 grams per day is beneficial for cognitive decline, and this is also true for wine. Okay. But no evidence or even detrimental effect for beer and spirit consumption. And you see there is a very nice meta-analysis of 29 prospective studies that was published a few years ago that found that mild alcohol consumption up to 11 grams per day, which is 1 alcoholic drink per day, is beneficial for cognition. And these are the graphs of meta-analysis indicating that up to 11 grams per day is beneficial for the risk for MCI or dementia, and again 11 grams per day only for dementia. And the authors separated their samples into 2 groups, older and younger adults, and you see that for older adults, sorry, I missed. And for older adults, okay, this is 17 grams per day, the cutoff, whereas for younger adults, it's 5 grams per day. And somehow this amount of alcohol was also replicated in the UK Biobank analysis, where you see that again cognitive performance is improved up to 10 grams of alcohol per day, 1 alcoholic drink, and then there is a deterioration. Specific for wine consumption, okay, there is another meta-analysis that also found that wine consumption is associated with a lower risk of cognitive decline. Okay, let's move now to coffee. Okay, again, no clinical trials, although they are easier in a sense compared to alcohol, but no clinical trials. And here, the evidence indicates that coffee is beneficial for cognitive decline. And again, the same meta-analysis that evaluated alcohol evaluated also coffee. And you see here that the cutoff is 2.8 cups per day. Okay, but if you go to older adults, this cutoff is to 4 cups per day, whereas for younger adults, there is no association found in this meta-analysis between coffee consumption and dementia or MCI. Okay, moving to fish. Okay, fish is well known for its omega-3 fatty acids, which is beneficial for brain. So observational evidence indicates this replicates these beneficial effects. No clinical trials, and this meta-analysis that was published a year ago under the title Meta-analysis of Observational and Experimental Studies found no experimental studies. So these graphs are only from prospective studies, and you see here that up to 2, let's say, servings of fish per week is associated with marginally non-significant risk of dementia, but if you go to Alzheimer's disease, there is a significant decline of there is giving association with reduced risk of Alzheimer's disease up to 1 or 1.5 serving per week, and then there is a plateau. Okay, meat, no effect of meat, okay, and no other studies thereafter. Okay, regarding fruits and vegetables, there's a lot of evidence about their beneficial effect, okay, and thereafter, after our review, only few studies about specific fruits and specific vegetables like berries and like green leafy vegetables, and also some reports that not only quantity but also variety of fruits and vegetables is important. This also was replicated in our studies. A paper that is in preparation, we found that sometimes variety is more important than quantity, because with a variety you reach a great amount of nutrients. And let's move to dairy. Okay, dairy is quite strange because you see here there are conflicting results from several studies, and under dairy we consider everything: yogurt, cheese, and milk, and possibly an answer comes from this meta-analysis, okay, that they were found that regarding the dose-response analysis and regarding the quantity of consumption, there's a non-linear association between dairy consumption and cognitive dementia, but there is an almost linear association between frequency of consumption. But the interesting thing is here, when these people separated the studies according to the continent, they found that, okay, for Asia, okay, the effect is, sorry, I cannot focus with this one, okay. In Asia, okay, there is a beneficial effect, whereas in Europe, no effect, okay. But you see here, one study in Australia found this detrimental effect of dairy consumption, okay. The authors speculate that maybe the quantity of consumption, of the type of the dairy, or the mode of consumption, okay. There may be several explanations about that. And let's move to seeds. Okay, there were no observational studies at that time, but since then there were 2 big observational studies, one on walnuts that found no association between walnuts and delay of cognitive decline. And this was also replicated in a meta-analysis thereafter. And the Singapore Chinese health study that found that the higher the nuts consumption, the lower the risk of cognitive decline later in life. And there are also 2 studies. These are the studies from the 2 cohorts of the Predimed study, but I will talk about them later on the Mediterranean diet, because these people did not only receive nuts, but also advice on the adherence to the Mediterranean diet. And let's move, and 2 RCTs, sorry, on almonds that found no effect. And move to olive oil, the hero of this conference, okay? Okay, actually no observational study that fulfilled our criteria at that time, apart from the one study that was published a few days ago, and we heard a lot about that study yesterday, okay? This was the study that combined data from the nurses' health study and the health professional study, and found that olive oil more than 7 grams per day is associated with more than 25% low risk for dying from dementia. And the other interesting thing is that when you substitute 7 grams of mayonnaise or margarine per day, you have this benefit from olive oil. And this was not true for other vegetable oils. But regarding clinical trials, we have these 2 studies, the Predimed studies, the 2 cohorts from the Predimed study, okay, and also 2 other RCT. One RCT that used a very specific early harvest high phenolic compound olive oil, and another one that evaluated the effect of Mediterranean diet versus Mediterranean diet and olive oil on cognitive function for 1 year, and they found that there was high reduction in cognitive scores in participants that were in the group with Mediterranean diet plus extra virgin olive oil. And I move now to the dietary patterns. I know that people who work with mechanisms do not like dietary patterns because they do not give important information for the pathophysiological mechanism. But on the other hand, I work with real people, with clinical practice, and we know that people do not consume isolated nutrients, isolated groups. They consume all this in combination, and all these nutrients have interactive or even counteractive effects. So for me, it's important to have the whole picture instead of specific nutrients or foods. And when we evaluated dietary patterns related to cognition, you see here that the winner is the Mediterranean diet. There's a lot of research about this dietary pattern. And cognition, there are many observational studies and also clinical trials, and the clinical trials are the 2 cohorts of the PREDIMED study. So I will briefly present, but I know that most people in this country are very well aware of this study. So the PREDIMED study, there are 3 groups: Mediterranean diet plus extra virgin olive oil, Mediterranean diet plus nuts, and the control group, the low-fat diet. And in one cohort, in the Navarra cohort, people evaluated cognitive function using 2 questionnaires, the mini-mental and another questionnaire, this clock drawing test, at the end of the intervention somehow. So we don't have change, but we have, in a way, only follow-up data. And it was found that Mediterranean diet and olive oil participants in this group and participants in this group scored better compared to the control group. But in the Barcelona cohort, evaluated cognitive function baseline and then at follow-up, and they have the chance to see also changes in cognition. And you see here that these 2 groups that were on the Mediterranean diet and olive oil, Mediterranean diet and nuts, scored better after the intervention compared to the control group. And I would say that global cognition perhaps is more important. And you see here that olive oil has participants in the Mediterranean diet and olive oil had a better performance. Since our review, there were many reviews and umbrella reviews and meta-analysis on Mediterranean diet. I have 3 of them that I like, but from all these, I very much like this table from the review of Gauci et al. That was published in 2022, because here you see there is a vote count of significant findings from each dietary pattern. And obviously, Mediterranean diet has the most significant positive results. So even these authors concluded that most of the available evidence supports the protective effect of the Mediterranean diet on cognitive function later in life. So what is the Mediterranean diet? Okay, the obvious question is the diet of people living in the Mediterranean basin. It was studied in 7-country study, but I would say that it was a study before. In the Rockefeller study that was carried out 2-3 years before the 7-country study, and because the food records was lost from the 7-country study, the only evidence about the true essence of the diet comes from the Rockefeller study. It's a plant-based diet, and I would also add it's also a diet of frugality. It's not a diet of abundance of food that contains mostly plant foods, legumes, fruits, vegetables, little fish and little dairy products, very little meat, and also olive oil as the main fat of the diet, and wine consumed modestly along with milk. There are many pyramids published worldwide. I like this pyramid that was published by the Fondazione DIETA Mediterranea because apart from the foods, their servings, their frequency, there are also other components of the Mediterranean diet like regular rest, conviviality, eating together, and on the other hand, eating seasonal local foods prepared with appropriate culinary activities. And in our research group in Athens, as we are part of the Mediterranean culture and the Mediterranean lifestyle, we thought what we can do to add to this huge research about Mediterranean diet. So our idea with Nikos Kermeas was to focus on lifestyle, the Mediterranean lifestyle that may be possibly more important than the Mediterranean diet. So we have a cohort there, the Heliot cohort. We collect lifestyle and medical and cognitive information from people who have a lifelong experience on Mediterranean diet, at least some components of this diet. So there are more than 1,700 people in two areas, in a representative area in a suburb of Athens and then in Larissa, somewhere in the middle of Greece. And these people, they go this cognitive examination, 2 hours of cognitive examination and all these cognitive tests. And when we evaluated individual food groups, we found that the pattern, the Mediterranean pattern, is more important than any individual food group. And we created this Mediterranean lifestyle index, combining not only diet, of course diet, but also sleep and also physical activity, and this component that was named instrumental activities of daily living, that for us has many activities that are well known for our country and for older people, like going to cafeteria, to cafe, playing cards, going to church, have discussion with local people in the streets and things like that. And we found that people who are in the upper quartile of the total Mediterranean lifestyle had more than 40% decreased odds of low cognitive performance. And when we evaluated the follow-up data, we found that even at 3-year follow-up, the effect is still there. So, by summing up, there are associations between nutrition and cognitive decline. However, the relevant research is challenging and the evidence sometimes is controversial, it's non-conclusive. I would say that there are several possible explanations, but for my view, 2 are the more important one. First of all, we do not know what is the exact time to evaluate or to intervene for cognition, because most of the people recruit participants here, somewhere here, where there is some cognitive deficit. For example, in our cohort, another cohort we have in Athens, the Albion cohort, with younger people, we found that these are non-demented individuals, okay? And we found that as you go to lower cognitive scores, but still not demented, people tend to move their dietary intake earlier in the day. So this is a connection with chronobiology and chrononutrition we heard yesterday from Professor Odorovas. And on the other hand, and now I'm going to give, let's say, the link with the following speaker. There are several mechanisms here. I'm interested in cognitive dysfunction, but the underlying mechanisms are many, and all these food groups are related to one or another, and sometimes they are positively related to one mechanism and negative to the other, so the net effect is difficult to be identified. In any case, I think that nutrition is an important modifiable factor, and I must admit that most of the evidence comes from the Mediterranean diet or the Mediterranean lifestyle. What is a plant-based diet, okay, with olive oil as the main dietary fat? Muchas gracias.
Thank you, Mary, for such a wonderful presentation, for being on time, which is per se an achievement. Well, it's a great pleasure to introduce the next speaker, which is a colleague, a friend, somebody I've been known for a long time. It's Professor Amal Kaddoumi. She's a professor of neuropharmacology in the Department of Drug Discovery and Development at Harrison College of Pharmacy in Auburn University. Dr. Kaddoumi has been really on the forefront of the basic research with a lot of translational approach in the area of what is, as Mary said, the start of the conference, extra virgin olive oil, and the different compounds of the olive oil. She did really important seminal work through her career, and she actually was very instrumental, even for myself, inspirational almost, because one of the things she tried to bring into the field, for example, was the vascular contribution of cognitive decline and dementia. So we go back to the cardiovascular, to the metabolic syndrome. So Dr. Kaddoumi, the stage is yours.
Good morning, everyone. And first, thank you, Dr. Praticò, for the nice introduction. And I would like also to thank the organizers of this conference for the invitation and for the excellent talks so far. Sorry. I am advancing. Yeah, someone can help me here. Yeah, I should go to the beginning. I think I will use that. Okay. So here we heard several presentations that talks about the observed effects of different of olive oil and maybe some of the phenolics in olive oil. And maybe for the research I focus on in my lab, it is more related to the question that Dr. Bizioli asked, which is the why. So while we are interested in the efficacy, but we are also interested in defining and identifying the mechanisms for the effects that we are seeing. So here, just kind of to remind everyone, when we talk about Alzheimer's disease, so Alzheimer's disease, it is, as we all know, it is memory dysfunction. And it is characterized by two main hallmarks, which are the amyloid plaques in the brain, as well as the neurofibrillary tangles. However, beside this, while everyone is focusing on these two hallmarks, but we have to remember also Alzheimer's disease, in the brains of Alzheimer's disease patients, you will find that it is also associated, you will find in neuroinflammation, oxidative stress, brain atrophy, that will collectively actually result in neuronal loss and eventually memory dysfunction. So in my lab, as Dr. Praticò mentioned, I focus more on the vascular dysfunction contribution to neurodegenerative diseases, and in this case, it is Alzheimer's disease. So as you can see here, and I hope it will work. Maybe this one? Oh, here is it. Okay. So this is here, which shows the, when it is in cognitively normal people, so you'll find the vessels, or what we call it, the blood-brain barrier, you also find that it is an intact and protective that maintain nutrients to the brain and also maintain homeostasis by removing waste products and supplying nutrients. Now, as in the case of Alzheimer's disease, and as it progresses from what is known as mild cognitive impairment to early Alzheimer, and then next stage of Alzheimer or severe Alzheimer with dementia, thank you. with dementia. So you will find there is a progressive dysfunction in the blood-brain barrier. You will find that it becomes leaky, where starts at the MCI stage, where the functionality of the endothelial cells of the blood-brain barrier, they become kind of not well connected. They become leaky in a way that it affects the transport or the movement of the waste product and the neurotoxins from the blood and the waste product of the brain, so that are not becoming, not efficiently removed and eliminated, that they will progress with the disease, and this eventually will result in beta amyloid accumulation and the parenchyma, vascular beta amyloid accumulation, as well as neurofibrillary tangles, in addition to other biomarkers that are associated with the disease. So now we are talking about extra virgin olive oil, and we have interest in understanding what is the mechanism. While there are many mechanisms, and I am sure about that, however, in my lab we focused on a few based on some of the hallmarks that we observed. So in this study, we tried to understand in preclinical study in animal models of our Alzheimer's disease, we wanted to answer what is the mechanism by which extra virgin olive oil actually improve the blood-brain barrier functionality or the vascular functionality, as well as reduce the pathology of Alzheimer's disease. So here in this figure, what we did here, we administered or added extra virgin olive oil to the animal's diet, mouse model of Alzheimer's disease, and for 3 months. And the age of these mice here, because we were thinking about the treatment mode, which means here in the case of we have the pathology already established. At the age of 6 months, we continued for 3 months treatment, and at the age of 9 months, we started to collect the data. So we collected the brains, and we found that because we focus more on the blood-brain barrier functionality, so as you can see in figure A, the green color here indicative of the extravasation of large molecules like IgG that should not be in the brain. However, due to the leakiness of the blood-brain barrier, you will find that there is more of this extravasation, and of course, this indicative of the dysfunctionality of the vessels in these animals. Now, with the treatment of extra virgin olive oil, as you can see in the lower panel here, you can see that the green color or the IgG extravasation is significantly reduced as a result of extra virgin olive oil. Now, of course, we will talk about one of the hallmarks in this case, which is beta amyloid. The green color is representative for the staining of beta amyloid, and what you will find that in the control group. And the control group here, by the way, is refined olive oil. We, afterward, now understanding more about the effect of olive oil or refined olive oil, I think what we are missing, or one of the limitations, is that we didn't, specifically with this study, we didn't have the wild type animals that should be the comparison, or the wild type in addition to Alzheimer's disease model that are not on olive oil completely. So here with the refined in extra virgin olive oil treated animals, we found that there was a significant reduction in beta amyloid levels, especially in the hippocampus, where the green color is significantly reduced, suggesting reduction in beta amyloid. So further studies with regard to beta amyloid, we found actually there are several mechanisms that have been enhanced and induced that are associated with beta amyloid, such as reduction in the production of beta amyloid due to inhibition of some of the enzymes associated with the beta amyloid production, as well as increase in the clearance and removal of beta amyloid from the brain that are either by enzymatic degradation or by transport or clearance across the blood-brain barrier. So in this slide, I also showed that we want to see the effect on neuroinflammation markers. So we did some experiments, immunostaining as well as Western blot, and we found that with extra virgin olive oil, there was significant reduction in astrogliosis or activated astrocytes, as well as we found reduction in some of the inflammatory markers like MMP9, TRPA1, and all these are associated with inflammation, as well as with some cytokines, as you can see here. Now, looking further for the mechanisms, we found that actually extra virgin olive oil here, it acted via suppressing the NLRP3 inflammasomes via inhibiting NF-kappa B pathway. In addition to that, we found that extra virgin olive oil enhanced autophagy, autophagy is important process in which beta amyloid is being degraded as well by the microglia as well as by the astrocytes, as you can see with the markers that been studied here. Now, collectively, with all these results, we found that, obviously, as expected, the memory function improved in the olive oil, extra virgin olive oil group. And in the next slide, I am summarizing kind of the pathway by which extra virgin olive oil improved memory, reduced neuroinflammation, as well as reduced beta amyloid levels and enhancing the functionality of the blood-brain barrier. Now, again, we have interest in the phenolic content of extra virgin olive oil. So what we did, we isolated oleocanthal. Everyone knows and heard about it. So oleocanthal, we studied oleocanthal, and again, in Alzheimer's disease mouse models. So, by the way, yesterday with that kind of the EVO testing, I was a little bit surprised. I'm not expert in extra virgin olive oil tasting, but if we felt any pungent taste, the pungent taste is coming actually mostly from the oleocanthal. Other phenolic compounds, they do have that pungent taste, but oleocanthal is the one that has the highest pungency. So here what we did, we administered oleocanthal, and I agree the dose is important. We studied dose-dependent studies for oleocanthal. Now, also, not only the dose, it is the matrix that is important. For example, when we refined or purified oleocanthal and we just dissolve it in saline and give it to the animals, we found that it requires higher concentration in order to produce the effect. So we saw at 5 milligram per kg, this is 10 milligram per kg. At 20 milligram per kg, there was effect, but we also found that there was hepatic toxicity, and this study, hopefully that will come soon. But it was not observed with the 10 milligram and lower doses. However, when we administered oleocanthal dissolved or suspended in olive oil, we found that it was effective at about 0.7 milligram per kilogram. So the matrix is important for the activity, suggesting that the importance of the fat of the olive oil, which I believe it is oleic acid. Maybe Dr. Sorry, is not convinced about that, but with the results we have that I'll show you, I am very convinced about that. So here again, oleocanthal, in the upper figure, you will find that the green color again for, the green color in the upper immunostaining graph. You will find that the green color is for the IgG extravasation, suggesting that you have a breakdown in the blood-brain barrier that was rectified with oleocanthal, as well as in the lower here figure, you will find that the green amyloid deposit was significantly reduced with oleocanthal, and that effect, besides, we did several mechanistic studies related to inflammation, related to degradation and beta amyloid production, and similar to the effect of extra virgin olive oil, suggesting that oleocanthal play some part in the collective effect of olive oil we observed in the previous slides. And this was also associated with improvement in memory function. Now, if we look, we are interested again, we want to see the effect on plasma levels as well as brain levels of beta amyloid 40 and 42. So as you would expect, I don't know. Okay, come here. As you would expect, the lower panel C and D, this is in the brain. Of course, this is the kind of comparison between the 4-month and 9-month of Alzheimer's disease model as 5xFAD, that we compare the beta amyloid levels comparison to wild type mice at the same ages. So as you can see at the 4 months of age, 5xFAD mice of beta amyloid 40 or 42 in C and D, you will see that there is higher level compared to the wild type. That with oleocanthal treatment in the green color, you will find that there is a reduction, significant reduction. And as the mice age at the age of 9 months, you will find that in the 5xFAD there is significant increase, again, as we would expect, that was significantly reduced in the presence of oleocanthal 10 milligram per kilogram. Now, interestingly, the plasma behavior is different than the brain. So what you will find here, in 5-XFAD at the 4 month of age, you will find that there is a significant increase in beta amyloid 40 and 42, and that was significantly reduced, a little bit higher than the wild type level, but significantly reduced with oleocanthal. However, as the mice age, imagine the CSF, kind of similar concept of the CSF. So you will find that because of the plaques deposition and in the brain, there will be less that is available into the blood. So you will find that there is a significant reduction compared to the wild type at the 9 month of age. That, with the treatment of oleocanthal, actually it increased, suggesting that is an increase in the functionality of the blood-brain barrier that can result in functional removal of the beta amyloid into the blood, which can explain part of the results, not necessarily the whole results. And actually, we were surprised with this data. Next, we wanted also to study the effect in the same mice, 4 and 9 months of age, 5-XFAD mice. We wanted to see and study the effect of oleocanthal in metabolic and behavioral phenotypes, changes in metabolic and behavioral phenotypes. So what we used here, we used the Promethean cages. Those cages are used in order to monitor the movement of the animals, the respiratory function, CO2 and oxygen consumption, and sleep pattern, food intake, and many different parameters that can. And why we have interest in that? Because in Alzheimer patients, you will find that they also are, their energy intake, their consumption of the energy is also, as well as a sleep pattern is affected. So we wanted to see the effect of oleocanthal in this case. So here, this is a summary of the results, and this is for the 9 month aged animals. So the dark blue is for the wild type, the dark red is for the 5-XFAD vehicle, and the green color is for the oleocanthal treatment. Collectively, what you will find here in general, that oleocanthal was able to restore the energy expenditure, that it was able to restore the movement of the animals, because animals, as they become, with progressive Alzheimer's disease, they become anxious, moving fast, and they consume a lot of oxygen, and when we talk about CO2, their levels. So actually, we can find that there is some restoration of these parameters in this case. So here, this is interesting data. We found that oleocanthal was able also to restore the sleep pattern, because these mice at 5-9 month age, they had less sleep hours compared to the wild type mice, and we found that oleocanthal was able to restore this function. Now, another interest we have is oleuropein. So we also studied oleuropein-rich, isolated from olive leaf extract. Similarly, we found that it reduced beta amyloid levels in the brain of the mice. Also, it reduced the neuroinflammation, and we were able to identify the mechanism. And here we can see that it improved memory function. And we tried to identify the mechanism by which oleuropein had such effect. And again, we found that oleuropein was able to reduce beta amyloid-related pathology by reducing neuroinflammation via suppressing NLRP3 inflammasome, as well as another pathway, which is RAGE-HMGB1 pathways via suppressing NF-kappa B pathway. Now, this is the summary of what we got from the results from collective data that we obtained during the maybe 14 years of my work with oleocanthal as well as extra virgin olive oil. So you will find that it was able to work on reducing different mechanisms like neuroinflammation, oxidative stress, reducing tau hyperphosphorylation, beta amyloid production, which collectively can result in memory function. Now, this next question was, okay, all this data is with animals. However, the next question was, does extra virgin olive oil prevent mild cognitive impairment conversion to Alzheimer's disease? So animals, whatever you give animals, it will work, to be honest. Yeah, I will not say that, I should not, but maybe this is the problem we have with Alzheimer drug developments, because in mice they are working, but when you move it to human, actually we don't see that efficacy. So the question was, okay, beautiful data in the animals, so what about in human? So for this study, we worked with MCI patients, mild cognitive impairment, and we designed our study to have two groups. One group with olive oil that is rich with phenolic content. We used the refined olive oil as a placebo for our study. And then we have them coming at the baseline. We did some cognitive function tests. We collected blood samples as well as feces samples for microbiota, but I will not show the microbiota results here. We did functional MRI and we did contrast enhanced MRI. And then we asked them, keep your diet exactly the same. We are not asking you to change any of your lifestyle. The only difference is add these 30 ml of olive oil that I am giving to you daily to your diet for 6 months. After 6 months, they came back and we repeated the same tests that we started at the baseline. So this is the inclusion criteria and exclusion criteria. The most important one here is that because for the MCI, that we selected those with a CDR score of 0.5, which is a characteristic of the MCI subjects. This table shows the baseline demographic and the clinical characteristics. There was no significant difference between the two groups, the refined olive oil as well as the extra virgin olive oil, with regard to the characteristics that are listed here, in addition to the cognitive tests results that are shown here, which are the MMSE and the CDR. So these results show the MRI data. So what we found in the functional connectivity, while refined olive oil didn't, the green color is after 6 months, and the red color is for the baseline data. So we didn't find significant difference between the post and before treatment, but with extra virgin olive oil, we found that the functional connectivity between the different regions of the brain actually enhanced and improved. In addition here, because remember, we are interested in the vascular function, so we found while the refined olive oil didn't affect the blood-brain barrier permeability, which is measured by the contrast agent we used, so there was no effect. However, with the extra virgin olive oil, while a significant effect, statistical effect, was not observed, but there was a trend toward reduction in the extravasation of the contrast. Now, the next question was, okay, let's focus on the hippocampus. Why the hippocampus were responsible for the memory. So we compared the permeability of the contrast in the hippocampus, and we found after 6 months of treatment with extra virgin olive oil, there was a significant reduction. There was a significant reduction in the permeation, suggesting enhancement in the functionality of the blood-brain barrier, while refined olive oil didn't show that effect. So now the effect on the memory function, you will find that refined olive oil, I think it's come. I thought it is you, Domenico, for the time. Tuck, tuck, tuck. It's not you, right? Yeah, okay. That was the agreement, actually, so. Okay, good. So here we found that for both refined olive oil and extra virgin olive oil improved the memory as measured by the CDR sum of boxes. We used another test, which is the WMS scale or memory function. And we found actually using the LM1 monitoring the LM2 as well as the LM2R that are explained on the other side of the slide, we found that both refined olive oil and extra virgin olive oil actually, they improved memory. And for example, LM1, or which is the immediate learning memory, there was improvement in 40% of the participants, 62% with the extra virgin olive oil, the same numbers with the learning memory 2 or delayed learning memory, and we found kind of about 50% in recognition learning memory improvement in refined olive oil as well as in extra virgin olive oil subjects. So memory improvements were there. In addition to that, we monitored changes in the beta amyloid as a biomarker and as well as tau in the blood. And we found that both refined olive oil and extra virgin olive oil actually resulted in significant reduction in the ratio of beta 40 to beta 42 to 40, as well as reduction in phosphotau ratio to tau ratio, suggesting that there are changes in the production, clearance of either of these two hallmarks. Now, we wanted to look at more specific on different pathways, what is being affected. So we did some omics study. This is the result from proteomics. So we found, and this represents the volcano plot of the proteomics data, and we found that there were several proteins that being altered by both olive oil, refined olive oil, and extra virgin olive oil. This slide here shows you what kind of pathways, proteins associated with what pathways that being altered in the case of the consumption of refined olive oil and extra virgin olive oil. So you will see the immune system is modulated, the lipid metabolism, coagulation factors, and other pathways were also altered by the consumption of refined olive oil and extra virgin olive oil. We also did some metabolomics analysis, and we were able to identify and see that there were several metabolites in the blood samples collected from refined olive oil subjects and extra virgin olive oil subjects were altered, and these metabolites play a role in, as you can see in the figure to the right, they are associated with the antioxidant effect, memory function, cholesterol metabolism, anti-inflammatory, in addition to other pathways that been altered because of the consumption of olive oil, refined as well as extra virgin olive oil. Now, what we observed, while both refined olive oil and extra virgin olive oil affected similar pathways, however, we noticed that refined olive oil effect was mainly higher in the following, in digestive-related salts that can help in digestion, as well as in regulating gut microbiota. We should not ignore the effect of oleic acid, in my opinion, that could play such role. With regard to extra virgin olive oil, we saw that there is more effect where increase in the expression of catechols, antioxidants, and essential amino acids that are important for neurotransmitter synthesis, as well as for neuro protection. Now, currently we are in our second clinical trial, and in this trial we are trying to answer the question, does extra virgin olive oil, you can see the map now, which is here, does extra virgin olive oil induce gene and metabolic changes in healthy subjects with Alzheimer's disease family history? So we are recruiting those with family history, and but currently they are healthy, in order to understand whether olive oil will protect them, and if it does, by what mechanism. And that's why for this study we are doing some omics data, collecting blood samples for omics data, that in collaboration with Dr. Praticò and Dr. Tassos and Basilis, that we are working on this study. So, of course, at the end, I will like to acknowledge for sure the students who do most of this work, as well as my collaborators, as well as the funding that supported this work and other work. And thank you, everyone, and available for any questions.
Thank you very much. Such a wonderful presentation. And I swear it wasn't me doing that noise. I wasn't making that noise. Yes. Because we had a little agreement with Dr. Kadumi that the timing was important. At the moment she would reach her time, I would start doing this. But I promise I wasn't me. It was somebody else in the back. They must have heard us doing that. Anyhow, so very exciting. The session is now open for questions. I think we have time for 2-3 questions. Anybody in the audience? Please. Microphone, one second.
question for Dr. Yannakoulia, because I think it's a bit surprising that the MIND trial failed to observe any effects of the MIND diet on improving the cognitive function. However, the Praticò trial is so different. So I'm wondering, I just want to pick up your brain regarding why we observe very different findings between the Praticò trial versus the MIND trial. Of course, the MIND diet, if you think about it, it's a hibernation. It's a hybrid between the DASH diet and the Mediterranean diet. But I just want to ask your opinion regarding how to interpret the difference between the MIND trial and the Praticò trial. Thank you.
Okay, actually this is a difficult question because if you saw in my slide, there was also a good evidence, epidemiological evidence for MIND diet. And actually the essence of this diet, it was invented exactly for this issue, for the prevention of cognitive decline. And I think all scientists were quite disappointed about this null effect. Okay. Okay, there are several explanations. Okay, one explanation is that it was the intervention was not for a significant time period. So, or this is one intervention, and the other intervention is that you see, human beings are quite complex. So we do interventions, and we think that we advise people to follow Mediterranean diet or MIND diet, and these people follow some aspect of the diet, but not the whole diet. And on the other hand, it doesn't mean that if these people add, let's say, olive oil in their diet, it's only that. They may also have some other changes in their diet that have not the beneficial or a detrimental effect. So, I mean, from my point of view, I'm a nutritionist. I would like to go back and see what exactly these people used to eat during the intervention, first of all, and then to extend the time of the intervention.
Thank you very much.
Any other question from the audience?
So thank you for these very interesting and nice presentations. I have a question for Dr. Kadumi. So in the trial you conducted, you asked your participants to change only the olive oil consumption, but to keep the rest just as it was. But we have heard in previous talks that just the consumption of extra virgin olive oil may promote some other changes in diet, because some other changes in diet, such as a higher consumption of vegetables, eventually lower calorie intake. So did you monitor the diet of your participants? Did you see any changes in there besides the olive oil consumption?
Okay, this is a very good question. And that's why in either this study and the current study, the study I presented and the current study, we have a nutritionist with us. So we monitor their nutrition at the beginning of the study and the nutrition at the end of the study. And we wanted to look at the data with interest, but at the end we found that there was no change in the pattern of the diet. So it was the same at the beginning and at the end. Yeah. So honestly for us, when we added only olive oil, because here we want to specifically answer the question of extra virgin olive oil. And remember, we are in America. We are in the south of America. When you talk about olive oil, it is rare use especially in the south of the United States. So that's why I do believe that they were sensitive to that effect, to be honest. Because we were really surprised to see it within 6 months, and with the memory function improved that level. So that's why this encouraged us to move forward and hopefully to answer the next question. And it will be interesting to see what kind of results we will get with those who are already on olive oil for long, and they consume it. Now, some of these people there in the south, they use olive oil and they don't care about what is the type of olive oil, only for cooking. So, and just kind of expose it to heat, cook with it, and eat it. And that's why I think we observed that effect. But with regard to the question, I will not say that's why, but at least I do believe that it played a role with that being naive to this, because maybe 2 or 3 participants, when they consumed olive oil and we asked them to consume it raw, you select whatever the method you want to use it, but don't cook with it, use it as you want. So some of them, actually, they had some stomach ache. They have some stomach problems, really, that level. So, and then we asked them to change instead of drinking it, for example, several of them drank it. We asked them to just mix it with food. And some of them, they prepared it with a smoothie or with the protein shake. And so they include it in their diet. So it just disappeared, that effect. But nutrition, with regard to your question, nutrition didn't change. Thank you. Sure.
There is an urgent question. Somebody who's dying to ask something. Please do now or never. We have 1 minute left. No more question? Everybody's satisfied. Good, but we can still talk to them after the session. They will be available. So they will not run away, so they're still around. So thank you very much. So that's conclude the morning. Coffee is waiting for you. Coffee and food is waiting for you outside. Thank you very much. Let's stay here.